Hello, welcome to my presentation on Malathion. We'll be talking about environmental concerns and effects of acute and chronic exposure. First, we'll talk about Malathion's classification and structure. Malathion is classified as an organophosphate. More specifically, it is a acetylcholinesterase inhibitor. Chemically, malathion is a dithiophosphate malleate ester with a chemical formula of C10H15O6PS2. Acetylcholine is the primary neurotransmitter in all presynaptic ganglion and the only neurotransmitter in the postsynaptic ganglion of the parasympathetic nervous system. It is also involved in neurofetal development. Embryologically, acetylcholine is very involved in the development of the nervous system. It is expressed by developing neurons and during periods of axonal growth. Acetylcholine is synthesized in the neuroganglion in a single-step reaction with the combination of acetyl-CoA and choline. It is confined to vesicles while in the neuroganglion, and calcium uptake through calcium-gated ion channels causes the release of acetylcholine into the synapse. Acetylcholinesterase is synthesized in nerve, muscle, and hematopoietic cells. It's expressed from a single gene, chromosome 7. Function of acetylcholinesterase is achieved by a hydrolysis reaction where acetylcholine is cleaved into two parts by the binding of acetylcholinesterase acetyl group to the serine active site. It's broken into acetic acid and choline, which is taken back up by the presynaptic ganglion to be repackaged into new acetylcholine molecules. There have been several instances of significant environmental impact from the irresponsible spraying of malathion. Two instances of note were the aerial spraying of malathion in California and Massachusetts in the 1980s and 90s. The spraying was done to control the fruit fly outbreak in California and a mosquito outbreak in Massachusetts. In California, studies done to determine the effects of the malathion spraying discovered there was a drastic decline in the honeybee population as well as other non-target species being affected. In Massachusetts, it was found that there were massive fish kills attributed to malathion spraying, and the continuous spraying caused a delay in recovery of these non-target species populations. And to make matters worse, the environmental impact came full circle when some of the non-target species that were killed with the malathion were species that fed on mosquito larvae to help reduce the mosquito population. Malathion primarily affects the nervous system. In the peripheral nervous system, pre- and post-ganglionic effects cause multiple organ system problems. Signs and symptoms of acute poisoning are accompanied by a set of symptoms where the acronym SLUDGE is used, which stands for salivation, lacrimation, urination, diarrhea, gastrointestinal upset, and emesis. This is caused by the overstimulation of the parasympathetic nervous system. In the central nervous system, malathion seems to mostly affect the hippocampus and the corpus stratum, which causes short-term memory loss, oxidative stress, and neuroinflammation. Treatment for acute malathion poisoning is accomplished through a two-drug combination. First, atropine is administered, which is an anticholinergic, which binds to receptor sites, interrupting the massive parasympathetic response caused by the inactivation of acetylcholinesterase. Atropine only works on muscarinic receptors, therefore the drug pralidoxime is given. Pralidoxime acts by having a higher affinity of being phosphorylated by the malathion molecule. This causes a competitive antagonistic action and acts on nicotinic and muscarinic receptors. Chronic malathion exposure is seen primarily through neurodegeneration symptoms caused by redox stress from non-cholinergic mechanisms and caused from the production of chlorpyrifos, which is an irreversible acetylcholinesterase inhibitor and has connections with the cause of Alzheimer's disease. Malathion also has effects on the immune system. Although the process is not well known, there are three possible mechanisms for this. Granular exocytosis affects cytotoxic T cells and natural killer cells, the induction of apoptosis, and serine hydrolase activity. Since the 1950s, malathion has been used safely not only in commercial and household settings as a pesticide, but for medicinal use against organisms such as lice safely. And malathion continues to be a safe pesticide and medication when used as directed. But we do have to take caution when using it irresponsibly in situations such as California and Massachusetts, where area spraying causes death of non-target species and causing serious environmental impacts.